All right, so we're going to finish off on photosynthesis and get right into genetics. So I believe we left off this slide here. All right, so just a quick review. We're talking about the light reactions here, photosynthesis, and then I think we already talked about no, we haven't talked about the Calvin cycle. Yeah, we're not to the Calvin cycle yet. So these are the light reactions still, right? So we have two photosystems. One, we're going to excite the pigment, the chlorophyll pigment. This is going to bump that electron up into a higher energy level, and then we're going to use this to make ATP. Okay, and that's kind of what you need to know there. From 1 to 2, the excitation of the initial pigment, right? First photosystem. Right, that electron gets excited, you move into a higher energy state, and then we can use that energy to make ATP. Okay, this ATP will then be utilized later on in the Calvin cycle to fix carbon to make glucose. Okay, that electron will then fall down the electron transport chain. Right, you have different cytochromes here, different enzymes that are going to facilitate that. The second photosystem, again, more light will be needed, and then it'll further excite that electron up. And then these electrons will then be passed off to NADP, okay? And remember, this is different than NAD in the mitochondria, okay? NADP is going to be reduced, so electrons are going to be added, and then it's going to be turned into NADPH, right? So you're going to have these electrons added, and this is what we call reducing power, or what I like to call the building blocks to your carbon molecules, okay? So we do have NADPH and NADP in our cells as well. Bacteria utilize this as well. NADPH is used to build, okay? So this is gonna be reducing potential for building organic molecules, right? So when you build organic molecules, you need to have energy, and you need to have some sort of source or some sort of component to add to those molecules in order to stick things together, okay? And these electrons are going to essentially provide that source of glue, or that source of reducing power that's needed to build the carbon molecules, okay? So think of these electrons as something that you're going to add later on into the equation to stick carbon or carbon dioxide together in order to make your sugar molecules, okay? So this is almost like the Lego pieces that you need to make the big model, right? This is the energy from photosystem 1, and from photosystem 2, you have your building blocks, okay? And that's really what you need to know. I don't need you to know any of the nitty-gritty stuff here. I need you to know the big picture stuff, right? NADP turns into NADPH after the electrons get passed off to it. These electrons will then be utilized to carbon fixate, right? To grab carbon dioxide. This energy will be used to grab carbon dioxide, and then this NADPH will be used, this electron will be used, these electrons will be used to stick those CO2 molecules together in order to make glucose, okay? And that's essentially what you need to know, all right? There's two photosystems here that get excited by light. They get excited, so they shoot this electron up into a higher energy state. I know that's kind of hard to think, but these photosystems do have certain metal ions in the middle, but I believe magnesium for both, and they vibrate when light hits the photosystem, and then they can actually, the energy going to be transferred to those electrons, which can be passed down to this ETC, or it can be passed off onto NADP, and then convert that into NADPH, okay? So again, photosystem 1, energy needed to carbon fix, right, to make glucose, and then photosystem 2 is the glue, or those building blocks needed to stick carbon dioxide together, okay? All right, so how the thylakoid membrane works to convert light energy into chemical energy. Right, so here we have that photosystem 1. Again, this is going to help the electron transport chain, right? So these electrons are then passed between the membrane, and this passage of electrons is going to pump protons from outside of that membrane to inside of this thylakoid space, okay? And this is going to make this fairly acidic, right? And here we have a potential now, right? So think of the dam and the water. This is potential energy. Okay, so this is just like the mitochondria, right? The inner matrix of that mitochondria. You have the acidity and the increase in pH. And then you have ATP synthase that can then be utilized for converting ADP into ATP, right? So this potential, think of it as that water being held back by the dam. And here we have the dam, which is the ATP synthase. Again, this is how we're going to harvest the ATP needed to go into the Calvin cycle in order to carbon fixate, okay? So this is essentially what photosystem 1 is used for, right? After these electrons get passed down through the electron transport chain, right, the electron transport chain is going to be actively pumping in hydrogens. The electrons can then go to photosystem 2, get excited, and these electrons will then be passed off onto NADPH. Again, NADP, sorry, and then that goes into NADPH once the electrons are accepted. Once this occurs, this molecule can then go into the Calvin cycle, again, utilizing that ATP from the ATP synthase, right, this energy that we just got from photosystem 1. And these two, the energy will then go to the Calvin cycle, as well as this reducing potential that we have in NADPH, which again, think of it as the glue that's going to be added to the different carbons in order to stick them together, okay? These are the building blocks that we need in order to make, or in order to gain our carbon source, or stick our carbon source together in order to make glucose, okay? So energy from photosystem 1 through the proton motive force, right? So protons moving, proton motive force. And then we get reducing power from photosystem 2. And reducing power, again, just think of NADPH as glue, okay? This is the glue that's going to be squirted onto the carbon molecules to stick them together in order to make glucose, okay? All right. If you have any questions, please let me know. I know this can be fairly confusing with all the different graphics, but those are really the two things that you need to understand about those photosystems. And the book provides these images, and I kind of don't like them, but you know, it might help some people understand this a little bit better. But that first photosystem, again, is going to be throwing these electrons up into a higher energy level, higher energy state, right? And then you can pass those down the electron transport chain, which will then pump protons into that thylakoid space, where then it can be used as a potential energy to pass through the ATP synthase, right? Then you gain your energy. Those electrons are going to be passed down to a lower energy state where they can be re-excited in photosystem 2 in order to dump off those electrons onto NADP to turn into NADPH. And again, NADPH, think of these electrons being housed in that molecule as reducing power or glue, right? This is the building block, the electron building blocks needed to make organic molecules, okay? All right. So the Calvin cycle, making sugar from carbon dioxide, and that's essentially what it is, okay? So we gain a lot of this, we gain energy, and we gain reducing power of the glue from the light reactions, right? This can be plugged into the Calvin cycle, where CO2 can also be plugged in, and this is going to be used to make sugar, all right? And that's essentially kind of the gist of what I want you to know there, all right? I'm going to go into the nitty gritty a little bit and might ask you a couple questions there, but for the most part, if you're explaining this on an exam, I just need you to talk about the light reactions and what we get out of it, okay? And how we get it. And essentially, we have the two photosystems, photosystem 1 and 2, that get excited. They get excited by light, okay? So light energy. And again, the electrons are going to be, one, for the first photosystem, are going to be used to make ATP. That second photosystem, you're going to get those electrons that are going to be picked up by NADP and converted into NADPH, and that can be used to make the glucose molecules, right? And so the outputs of the Calvin cycle are going to be the exact opposite of the inputs of the light reactions, right? So we have NADPH coming out. That makes sense because we're using the electrons on NADPH, okay? ADP is coming out as well as the phosphate. That makes sense because we're using energy in the Calvin cycle. ATP gets converted in adenosine triphosphate will be used and converted into adenosine diphosphate, okay? So again, very, very important processes that spit out those byproducts at the very end, okay, that go back and get recycled in the light reactions, right? So again, this is just a cycle that occurs over and over again in that plant cell or that photosynthetic cell, and again, continuously building those molecules, right? Also, the light reactions, we bring in light and water, and we off-gas CO2, or off-gas O2, or oxygen, right? In the Calvin cycle, we bring in CO2 and we get out sugar. All right. So yeah, we'll talk about it a little bit. I wasn't too sure if I should get into the details, but we should. All right. So there's going to be an enzyme that adds CO2 to the 5-carbon sugar called Rubisco. Okay. The resulting molecule breaks into 2 3-carbon molecules. All right. So Rubisco is going to be very important. This is going to be the most important enzyme probably in the entire world. And it's probably the most prevalent enzyme in the entire world. Okay. This enzyme that pulls in carbon and adds it to this 5-carbon sugar here. Again, this is probably one of the most important things you can do. Okay. Being able to grab CO2 and fix it onto another carbon source. And believe it or not, this is what drives the world. This is what makes the world function in terms of how we utilize energy and how energy is distributed throughout the surface of our Earth. Okay. So Rubisco is an incredibly important enzyme, right? Because it has the ability to grab CO2 and stick it on to another carbon molecule, okay? ATP is then added and phosphorylates the molecule. We have the glue from the NADPH being added as well. So using energy from ATP and NADPH produces a light reaction, and produced from the light reaction, enzymes convert each 3-carbon molecule into a 3-carbon sugar. G3P. Okay, G3P can then be used either to make glucose. Glucose is a 6-carbon molecule, or it can be used to make building blocks, or this is referred to as other organic compounds. Okay, so for every 3 molecules of CO2 that enter the cell, then the output is a G3P sugar, right? The other G3P sugars continue in the cycle, right? They make the 5-carbon sugar, 5-carbon molecule. All right. Using energy from ATP, enzymes rearrange the remaining G3P sugars to regenerate RUBP, and then Rubisco, again, is that enzyme that adds the carbon molecule to RUBP. Okay. So again, 2 turns gives you 1 glucose in this Calvin cycle. All right. What I need you to know from here is that we do have energy that's being used in this Calvin cycle. We also have the dumping of the electrons onto these carbon molecules in order to build or make more sugar. And that's something that you already had heard from the last couple of slides concerning the light reactions. Okay. So just know that, and also understand what Rubisco is. So I didn't, it's not really here. I should definitely make a slide. Sorry, I'll probably put something, a little note talking about Rubisco. But Rubisco is that enzyme that fixes CO2 onto the 5-carbon sugar here, and then it gets broken down by this ATP. So the ATP adding on the either end of that 6-carbon sugar here, again, that's going to be the important component in order to separate them out into G3P sugars. Okay. It's just like glycolysis in that regard, where when you break down glycolysis, you break down glucose in glycolysis, you phosphorylate either end of that molecule, and then it splits it in half. Okay. So important ideas about photosynthesis. NADPH is used as a building source for the cell. Okay, so remember, it's glue. Just think of it as glue. The electrons are needed to be added in that process in order to carbon fixate or make those glucose molecules. Okay. ATP is the energy needed as well for that Calvin cycle. Okay, so you saw multiple points where ATP is utilized to phosphorylate the different molecules. Again, we gain, or plants gain, that ATP from the light reactions as well. Okay, so plants can also break down their glucose molecules, right? We talked about starch as a storage molecule. That's going to just be repeating subunits of glucose. Again, plants can either gain their energy from the sun, or they can gain their energy from the starch that they store in their leaves and in their roots. Okay, so there's different mechanisms by which plants can gain their energy, right? They can perform glycolysis, they can perform respiration, cellular respiration, but this is how they are considered autotrophs, and this is how they actually make their own energy. And this is how they make the energy for the rest of the world as well. Plants make enough energy for themselves and us. So that's something to kind of really think about. These organisms have the ability to make plenty of energy for themselves as well as plenty of energy for the rest of the world. They're the building block, or the thing that sustains Earth and the life on Earth. Okay. CO2 is pulled from the air. Okay, so CO2 is brought in through the stomata, right, in those leaves or the little pockets or holes in the plant. That you can bring in CO2 and release oxygen into the environment. Okay, so NADPH needs to be recycled to NADP in order for the light reactions to work. If you have the light reactions have expended or filled up all of these NADPs into NADPH, but you don't have the Calvin cycle working, then you don't have the recycling, so the light reactions just don't work anymore. Okay, and that does happen in certain plants. And this occurs because sometimes you have the inability to open up your stomata during the day. The plants just won't do it because of the heat, because maybe they live in a desert environment. So they can shut down the Calvin cycle, right? They can stop bringing in CO2 because if they open up their stomata, those plants have potential of dying or becoming very, very dehydrated if they open up their stomata during the day. Okay, so they'll open it at night. And again, this is very common in desert plants, so cactus. So you do have to have the recycling of the NADPH to NADP in order to maintain the cycles from working in unison. And again, the light reactions and the Calvin cycle can happen independently of each other, right? So this is typical of cactuses, right? Cactus will perform the light reactions, store the energy, and store the NADPH. And at night, they open up their stomata, bring in the CO2, and start the Calvin cycle. But there's no light at night, so they're not running the light reaction. So they'll store up all this this glue or this NADPH, and then perform or carbon fixate at night, and then they're done. Okay, and this makes sense too because sometimes cactus or cacti don't grow as fast as other plants. All right. Any questions about that, please let me know. And we'll try to get through this as well. All right, so cellular reproduction, cells from cells. This is chapter 8. All right, so what is cell production and why is what reproduction accomplishes, okay? So cell division is the reproduction of a cell, right? So this is just making copies of other cells. And this is important for normal growth, maintaining of different structures in organisms, right? Also reproduction, okay? So there are organisms that are single-celled, like amoeba or certain types of protozoans, and they have to divide in order to make more of themselves, right? This is their form of reproduction. It's like asexual reproduction, or just roughly they're making clones of themselves, right? They're making two of themselves in this process. Okay, so chromosome, this is a gene carrying structure found in the nucleus of a eukaryotic cell, and most visible when compacted during mitosis and meiosis. Also the main gene carrying structure of a prokaryotic cell. Each chromosome consists of one very long thread-like DNA molecule and is associated with proteins. So it's important to note that chromosomes, there is such thing as called as a bacterial chromosome. but it's very different in structure in comparison to a eukaryotic chromosome. Okay, so DNA and bacteria are there's something called a supercoiling that's performed. So it's when the DNA is almost wound up really, really tightly in a ball. Okay, and it's like twisted together. So that what I like to think of this form, or how this looks or how this occurs, is think of old school telephones, and they have the coiled wire. So if you've ever dropped the phone or somehow got the wires really tightly spun up, and you try to pull it apart really fast, sometimes the wire will ball up fairly quickly and just get in a tight knot. And that's exactly how bacteria actually organize their DNA in a way. They do something called supercoiling, or super, where it's very tightly packed DNA, but it's just balled up and just kind of, I don't know, it's like a towel. Think of a towel when you're wringing it out. It's all that, it's just getting balled up together really, really tightly. Okay. The way eukaryotes organize their chromosome is very different. They have something called histones. Those are going to be proteins that are going to have the DNA wrapped around in a nice organized manner. And then you have them organized in a chromosome and a chromatid. Okay. So 3 functions of cell division by mitosis. So again, we have cell replacement. So cells being replaced in the body. So growth and division, again, important in the body, right? As you get older, you need to start adding or becoming larger, depending on your life stage. So asexual reproduction as well. So we have reproduction of amoeba, fragmentation, and regeneration of a sea star. Excuse me. Reproduction of an African violet from a clipping, right? So it's like if you clip a plant and you stick it in another potting or another jar, you get the plant growth that way. So again, these are the 3 different functions of cell division by mitosis. All right. So again, this is just regular cell division. Okay. So what cell reproduction accomplishes? So we have asexual reproduction, the creation of genetically identical offspring by a single parent without the participation of gametes. So no sperm or no egg. So no genetic recombination. So sperm and egg, when I say genetic recombination, this means that you're having variations in genetics because you have 2 separate parents. Okay. You can have genetic recombination through asexual reproduction, as well as if you have a hermaphroditic parent where they have both male and female parts, or male and female gametes. And this can occur, but for the most part, when we're talking about just cell reproduction and asexual reproduction, we're referring to non-genetically recombinant offspring. So the offspring does not have or does not vary genetically from the parent. Okay. So clone is a collection of cells, organisms, or molecules resulting from cloning. Also, colloquially, a single organism that is genetically identical to another because it arose from a colony of a somatic cell. Okay. So a clone, again, just think of an organism that is genetically identical to itself, and when it reproduces or it divides, it's going to be genetically identical to that original parent or mother cell. Okay. So sexual reproduction is going to be the creation of genetically distinct offspring by the fusion of 2 haploid sex cells. So you have gametes, sperm and the sperm or egg, forming a diploid zygote. Okay, and we'll talk about haploid and diploid a little bit later. But sexual reproduction, just think of the whole point of sex and the whole point of sexual reproduction is genetic diversity. So, and why is that important? Well, genetic diversity, allows for evolution, allows for survival in differential environments. So differential environments meaning that there are going to be different changes in your environment that might support life better for a genetically different organism, right? Let's say I survive better in colder temperatures and someone survives better in the desert, right? And this inherent advantage might be provided through genetics, okay? Not through environmental stresses, not through training, or not through conditioning by parents. This can be solely provided by the fact that my genetics are just a little bit different than that other person's genetics. I can survive better in the heat. right? And this variation in the population is incredibly important for survival for certain organisms, right? This is how bacteria can eventually become resistant to antibiotics, okay? Right, some bacteria are just more resistant to antibiotics than others, and those will survive after the antibiotic chemotherapy. This gives them an inherent advantage. And again, sexual reproduction is going to be all about that variation, right? All about the inherent differences that need to be in a population, because if every single one of those organisms is susceptible to the antibiotic therapy, then they will all die and they will become extinct, right? So those variations among that population is what's important to allow that organism to survive, all right? All right, so gametes. So sex cell, a haploid egg or sperm. The union of two gametes of the opposite sex is fertilization, produces something called a fertilized egg, which is a zygote. Okay, so a zygote is a fertilized egg. Once the sperm meets the egg, boom, you have a zygote. All right. So chromatin, histone, and nucleosome. So chromatin, this is a combination of DNA and proteins. So those proteins that I was talking about earlier, the histones, we'll see in a minute, this constitutes a chromosome, okay? Often used to refer to very extended form taken by the chromosomes when a eukaryotic cell is not dividing, okay? So typically what we see is this DNA is going to be packaged up and stored DNA, okay? So this is not going to be the DNA that's actively being transcribed, okay? So histone, this is a small protein molecule associated with DNA and important in DNA packing in the eukaryotic chromosome. Again, this histone is going to be used to make chromatin, right? So this is going to be the DNA and the protein component, so the histone. Nucleosome is a bead-like unit of DNA packing in a eukaryotic cell, consists of DNA wound around a protein core made up of 8 histone molecules, right? And we'll see that in a minute. So organisms also have various amounts of chromosomes in the cell body. Somatic cells. So somatic cells are technically body cells, are just your normal cells that are not your gametes, okay? So in humans, as we know, we do have 46 chromosomes. We have 23 pairs, right? So 46 different chromosomes, but you have 23 homologous pairs. And what's that mean? So you have one chromosome from or chromatid chromosome from your mom or your dad, and then they come together to make that homologous pair, okay? But each of those chromosomes contains the same, even though they're homologous pairs, they have similar genes or the same types of genes on each chromosome, but they have variation or differences in each, because one comes from your mom, one comes from your dad, okay? So again, there's 23 pairs, 46 different chromosomes, okay? So half from your mom, half from your dad, okay? But here you have a koala with 16, you have a giraffe with 30, you have this rat here with 102 chromosomes, a dog with 78, bison with 60, right? So we do have organisms that have more than us. It doesn't equate to genetic complexity in any way in terms of the amount of genes or the amount of intelligence or strength, or these extra chromosomes don't equate to anything, right? They're just separate. Separate components of the genetic information that may produce the same amount of protein or same amount of genes, and they really don't equate to anything more than that. It's just how they're organized in that specific organism. Believe it or not, the chromosomes are actually a way for to prevent breeding between species. Okay, so it's actually hypothesized that the differences in the amount of chromosome will allow or prevent interbreeding between different species. Okay, and this can be bad, right? You can still breed certain organisms together, right? There's like a horse and and a zebra can breed, a donkey and a zebra, right? But they don't produce viable offspring. And in partially because of this reason, right? They may have different or varying amounts of chromosomes that will not allow for their gametes to form properly, okay? And we'll get into how that occurs later on in this lecture. But again, it's a protective mechanism. So here we have colored chromosomes. This is a plant cell just before division. Again, these chromosomes are colored with stain. All right, so you can see this organization of DNA with protein very nicely here. And this is going to be, this looks like it's in anaphase, or no, telophase. Maybe this is forming mitosis. But yeah. Actually, this looks like telophase. Probably, it says cell division right before. All right, so yeah, it's probably telophase. So this is going to be ready to split in half right here. So in plants is a little bit different because they do have a cell wall. So they'll form a cell plate here right in the middle, and then they'll have the splitting event occur. So they'll separate out into two different cells that way. Okay, so here's a nice representation of a chromosome. So here you have sister chromatids, right? These are going to be copies of each other. And so this cell is getting ready to probably start cell division just because there's two sister chromatids. So copies have been made of your homologous pairs, them individually, right? So here we have the histones and DNA, which makes up a nucleosome. So 8 of these proteins wrapped around with DNA makes a nucleosome. And then you have the chromatids. This is going to be referring to that DNA and protein packed together. And again, histones and the DNA make up the chromatids. All right, so duplicating chromosomes. A cell must be able to duplicate its chromosomes before it can divide, right? So sister chromatid, one of the identical pairs of a duplicated chromosome. While joined, two sister chromatids make up one chromosome. Chromatids are eventually separated during mitosis or meiosis II. The centromere is a region of chromosome where two sister chromatids are joined and where spindle microtubules attach during mitosis and meiosis. So here we have the centromere here. And again, this is going to be an area where these chromosomes are attached to by those microtubules. Excuse me. And when these microtubules attach, they're actually going to attach and rip apart those sister chromatids in order to separate in their two new cells. Okay, and in the centromere, you do typically have what is referred to as junk DNA or unimportant DNA because this area is prone to damage just because of the attachment site of the microtubules. All right. So during cell reproduction, the cell duplicates each chromosome and distributes the copies to the daughter cells, right? So think of this as the initial one single cell. You have the DNA and then it duplicates, right? This duplicate is going to be a copy of one of those 46 different chromosomes, okay? Right? You need to make copies of both your mom and your dad's homologous pair, right? So you have 23 pairs. So one for your mom, one for your dad, right? Each of those need to separate out and then make copies of themselves. And then they can donate into their respective cells, right? So this is one, again, chromosome. So this is either from your mom or your dad. You have another pair that's from your mom or your dad that's going to be performing the same process, right? So this will be duplicate. This will be the exact same, right? These are copies of each other. You have sister chromatids now, and then they can separate into their individual cells once the division occurs, okay? So I know that's really confusing and that's probably going to confuse a lot of you when we talk about this and we test. Think of just the human model for the majority of this, and this is kind of what I'll use. I won't use like a dog or a bison or anything like that. I'll be mainly referring to human chromosome counts, okay? So the 46 different chromosomes are going to be referring to the individual chromosomes that you get from your mom and your dad, right? Whenever we talk about 23 homologous pairs, right, you get one for your mom, one for your dad that are analogous or that are similar to each other, right? So let's say mom donates eye color, dad will donate another chromosome with eye color. Those two will come together. And of course, either the dominant gene will take over and show that eye color, but they're different chromosomes, right? That house two of the same genetic information, right? So they both will have eye color, digestive something, your allergies to gluten, I don't know, doesn't matter, right? But they're going to have the same genetic information, and they're going to come together to make up that one homologous pair, okay? But they're separate. During this event of chromosome duplication, these homologous pairs will have to make a copy of each other, okay? And they need to separate again in order to go into their respective cells, okay? So I know that again, that's a little confusing. If you have any questions, just stop and let me know if how I can make that more sense of that explanation, okay? But again, yeah, it's a little difficult concept to kind of grasp, just because you go from 46 to 23 to 46. Technically, at one point, you'll have, I think 90, what is it? 96, 46, 80, 90, 92 pairs of copied chromosomes. So you have the 46 original plus an extra copy of each of those 46, because they're going to be donated to another cell during cellular division, okay? Which is like crazy, right? It's a lot of genetic information in one cell, okay? All right, so the cell cycle is an ordered sequence of events, including interphase and the mitotic phase, that extends from the time of time a eukaryotic cell is first formed from a dividing parent cell until its own division into two cells. So interphase, the phase in the eukaryotic cell when the cell is not actually dividing. During interphase, cellular metabolic activity is high, chromosomes and organelles are duplicated, and the cell size may increase. Interphase accounts for 90% of the cell cycle. Excuse me. So this cell cycle, again, it's incredibly important for that cell to stay organized and to stay within a certain parameter of growth, okay? And this is incredibly important because it prevents cancers from forming, okay? So the reason why we have so many different control points, not only is it because of the complexity of our cells, but also we want to make sure that we're not going to grow any cells that are going to be potentially cancerous, okay? So we do have check systems, right? Interphase, again, is going to be the majority of the time spent. The cell is going to be spent in 90% of the time, and then 10% of the time, that cell will be dividing. Excuse me. And this is also dependent on the type of cell, right? There's certain cells in the body that actually do not divide at all, like nerve cells, and there's cells that divide all the time, such as epithelial cells. So depending on the type of cell, where the cell is found, will heavily determine whether or not interphase percentage in interphase varies, okay? So mitotic phase, or the M phase, the phase of the cell cycle when mitosis divides the nucleus and disrupts its chromosomes to the daughter nuclei, and cytokinesis divides the cytoplasm, producing two daughter cells, okay? So we'll talk about M phase, and we'll talk about the various components in it. So this is mitosis, or referring to mitosis. So you have, you have interphase, metaphase, anaphase, telophase, and cytokinesis, right? Interphase is going to be that separate phase. It's going to be away from the mitotic phases, or the M phase, okay? So cytokinesis is the division of the cytoplasm to form two separate daughter cells. Cytokinesis usually occurs during telophase of mitosis, and two processes, mitosis and cytokinesis, make up the mitotic M phase of the cell cycle, okay? So this will be that last event that separates the two new daughter cells into their own units, okay? So this is that pinching, the invagination of that membrane, and then that separation between those two cells, okay? Do I have a better slide? Oh no, I don't have a better slide. Okay, this is it. All right, because this is kind of hard to see. I'm sorry. So cell reproduction, this is again referring to prophase, metaphase, anaphase, and telophase. Okay, so prophase, this is after the duplication that occurred in interphase, right? So all those chromosomes have duplicates now. Here you have the nucleus is going to start to break down. You have the initial forming of the mitotic spindle. So these are those microtubules that are going to attach, these little outbranching microtubules that are going to attach to the chromosomes at the centromere. Okay, so remember the centromere is going to be that center of that chromosome where there's a little bit of junk DNA, which means that it can take some damage there. And this can be a very damaging event because think about it, these microtubules are reaching out, grabbing those chromosomes, and then ripping them apart from the centromere. Okay. So again, prophase, think of the nucleus breaking apart. Think of the organization of these mitotic spindles with the microtubules reaching out and grabbing at the centromere, at the center of those chromosomes. Okay. Metaphase is going to be the phase where these the mitotic spindles line up down the middle. There's going to be a line of these chromosomes, these condensed chromosomes at the center. Here's a nice little image of that. So it's just going to be the nice organization down the middle to separate into the eventual two halves of that cell. Okay, so microtubules again are attached at the centromere. Anaphase again is going to be that pulling apart of those individual chromosomes. They are now separated. So each copied pair is going to be in its individual cell, quote unquote, because it's not technically individual cells yet. This is still one cell because there hasn't been a separation. But again, they're pulling into either end of those two cells, so you have the genetic information conserved in each cell. Okay, so that's anaphase, the pulling apart after metaphase. Telophase, this is going to be the nucleus starting to reform, the mitotic spindles going away. You have the invagination, or this is called the cleavage furrow, or you can call it an invagination of the cell membrane. So this is going to be that pinching or the closing off of that membrane in order to separate into two individual cells. So telophase is going to be that the forming, everything's kind of coming back together. Cytokinesis is what we refer to as the splitting of those two cells. Okay, so cytokinesis will actually be the splitting or the separation of the two, not telophase. Telophase is a start of that. So telophase is your nucleus is coming back together, your DNA is separate. Now you're losing the microtubules and the centromeres. And now you're getting ready for cytokinesis. But cytokinesis is going to be that separation of the two cells specifically, not telophase. Telophase is the beginning and then the reforming of the nuclei. Okay. So prophase, again, first stage of mitosis, duplicated chromosomes condense to form structures visible with light microscope, and the mitotic spindle forms and begins to move the chromosomes toward the center of the cell. Excuse me. Metaphase, the second stage, the centromeres of all the cell's duplicated chromosomes are lined up along the center line, right? It's going to make that metaphase plate. Anaphase is going to be those chromosomes or chromatids being pulled apart and kind of forming in each separate end of that cell. Telophase nuclei again starts to form. Two poles of the cell are seen. And telophase then will occur together with cytokinesis. And cytokinesis again is after that pinching of the membrane. It's going to be that separation is referring to that cytokinesis. Okay, so here we have the cleavage furrow. This is showing a nice animal cell that's going to be separated out into two daughter cells. So that cleavage furrow or that invagination of that membrane again is going to be that start of anaphase. And then eventually cytokinesis, okay? Or that splitting. The cytokinesis is referring to that split, those two cells. All right, so in plants, it's a little bit different, right? A cell plate needs to form because of the cell wall, okay? So remember the cell wall structure is made from cellulose. Cellulose has all those cross links and that very strong, rigid organization of that cell or that cell wall. So it's very difficult to separate, right? You can't just pinch that cell wall in half and separate two plant cells, right? You need to make something called a plate, right? A cell plate, which is going to be cellulose as well, but this new plate is going to then separate the two nuclei that were created from the daughter cells, okay? Excuse me. And this plate again is gonna grow in the middle and in between and separate out those two nuclei, and then eventually you'll have the two separate cells. Okay. So cancer cells dividing out of control. So cell cycle control system. This is a cyclically operating set of proteins that triggers and coordinates events in the eukaryotic cell cycle. Okay. So I guess that's not that important. Just think of this as kind of like the operating system of what triggers cells to divide and not divide, okay? Um, and again, this is very, very important for tumor or cancer control, okay? So a tumor is an abnormal mass of cells that forms within otherwise normal tissue. A benign tumor, an abnormal mass of cells that remains at its original site in the body. So this is has not moved or metastasized into other areas, okay? Um, metastasize just means that it's moved out to another part of the body. A malignant tumor, abnormal tissue mass that separates into neighboring tissue and to other parts of the body, a cancerous tumor, okay? Cancer is a malignant growth caused by abnormal uncontrolled cell division and then metastases, the spread of cancer cells beyond their original site. So it's very important that you kind of differentiate the different types of tumors and different kinds of cancers, um, and kind of understand what cancer is. I know that we haven't really talked too much about cancer, but when we talk about the cell cycle, we need to, right? Because the cell cycle is in place and the cell control systems are in place to prevent the overgrowth of cells, right? You do not want this to be occurring, right? Nonstop in the cell. Um, this can provide problems, especially if you have uncontrolled cell growth. You might have inappropriate separation during anaphase. Ooh, excuse me. You might have increased mutation if you do have a cancer cell line that's going to start growing, and eventually you'll have the forming of the cancer, right? And this cancer will take the energy away from your other cells. It'll outgrow your other cells or your other tissues, and then this is where you have the detrimental side effects of having a tumor, right? And again, these cells and these tumors are dividing at such a rapid rate. They have even a different metabolic profile than the cells throughout your body, right? They're going to be increasing the amount of glucose usage. These cells actually have a higher temperature. So even there have been cases where women have walked through, or men even have walked through heat detectors, and you can see the tumor light up the heat detector because the cancer cells are just dividing at such a rapid rate. They're causing so much heat to be given off of that tumor. And again, you can even see that in some thermal imaging devices. Okay, so I know that there's been a few cases of women that have been told like, hey, there's a hot spot on your chest, and they find out that it's breast cancer. Right? There have been people with thyroid cancer that have growths they didn't know about, and on a thermal detection device, they were able to see like a hot patch on their neck that turned out to be a cancer. Okay. So growth and metastasis of a malignant tumor of the breast. Okay, so if you have a breast tissue with a tumor, eventually it can turn to cancer. This cancer can then metastasize, which means it spreads to the lymph vessel, the lymph node. Okay, and this is a terrible problem, right? You do not want breast cancer or cancer in general to spread to the lymph nodes and then disseminate throughout the body. Because if that occurs, now you're treating that patient systemically rather than in that one spot, right? So if you have surgery to remove the tumor here, you can probably even save the breast at this point, right? You just cut the tumor out and pray that it doesn't grow back again. Whereas if you're here, it doesn't matter if you have a double mastectomy at this point or remove the whole breast tissue. This cancer has disseminated through the blood and your lymph vessels. Now it's a systemic problem, and now you need to treat the person as a whole, because you can't just remove uh, uh, the body's lymph system or blood vessels, right? There's so much cancer that's disseminated throughout. It's a problem. Okay. So radiation therapy or cancer treatment. So treatment for cancer in which parts of the body have cancerous tumors are exposed to high energy radiation to disrupt cell division of the cancer cells, right? So we also have chemotherapy. So this is treatment of carcinogenic or uh, drugs and that disturb cell division. So these are actually toxic. So both of these um, forms of treatment are very, very toxic, right? So you hear people that undergo chemotherapy and they lose their hair, right? And it's because these chemotherapeutic drugs will actually destroy or prevent those cells from dividing at normal rates in your body as well. Okay. Um, Cancer cells divide at a much higher rate. Therefore, you treat them with these drugs, and essentially what happens is you can destroy or disrupt those cancer cells because of their how fast they divide. But these drugs will also affect the cells in your body that naturally divide rapidly, such as your epithelial tissue, so your mucous membranes, your hair follicle, your hair tissue, or the tissue that grows your hair, right? Your follicles. You'll start losing your hair because again, those chemotherapeutic drugs will stop that tissue from growing normally or naturally, as you would see with individuals that don't have the chemotherapeutic drugs or that do not have cancer, right? And again, this radiation, this chemotherapy essentially is exploiting or taking advantage of the cancer cells growing at a much higher rate than the other cells in the body, right? But there are new treatments, which a book didn't mention, which I was really surprised about, such as immune therapy and hormone therapy, right? Um, so individuals that have like prostate cancer or breast cancer, you can have hormone therapy. So um, I know we talked about steroids a little bit, but like we talked about the androgenic side effects. So testosterone does have a negative, well, I wouldn't say negative effect on the prostate, but it does cause that prostate to grow. So it does cause that tissue to grow. And same thing with different sex hormones in females as well. You gain your secondary sexual characteristics um, um, after your pubescent years, right? So, and same thing with males. You have the darkening of the genital areas. You have the pubic hair. Women have enlarged breasts. Men will develop different musculature. So these hormones actually can dictate how certain cancers grow, because if, especially breast cancer or prostate cancer, they're found in uh, um, our specific sexual organs, or even testicular cancer as well. Uh, you can control it with hormone therapy, especially if the tissue is sensitive to hormone control or hormone or sex hormones. Okay, so they talk about breast cancer having either the HER receptor, right? They talk about hormone therapies that help certain types of breast cancers as well. And again, this is going to be using hormones in order to stop or slow the growth of that breast cancer, right? And you hear of triple negative breast cancer, and this is going to be breast cancer that lacks the receptors for a couple of different hormones, as well as the HER molecule as well. So triple negative, the HER2 receptor is actually gone, so you're not able to control that breast tissue with the different hormonal therapies, right? And this can be kind of scary. Triple negative breast cancer is probably one of the more scarier cancers to get, besides like pancreatic cancer, which is like really bad. You don't want pancreatic cancer either. The prognosis is very, very bad. But triple negative breast cancer is up there as well. Um, just because you don't have those hormone therapies that are available that other women would have. Immune therapy is also a new kind of a new idea where they're actually taking immune cells that are found in the tumors and extracting them. And so what happens is they extract these immune cells, and they're your own immune cells, from this tumor, and then they're going to grow them in a separate unit in the hospital or some lab, right? And then what happens is they then transfuse those immune cells back into your body at higher numbers, and hoping that these immune cells will target the cancer that's systemically spread around through your body, okay? And they've had major success with this in skin cancers as well as lung cancer. So very, very, very promising work being done with immune therapy. And again, it's just utilizing your own body's immune system to fight off cancer, which is part of the reason why we have a cell cycle and part of the reason why we do have an immune system, right? It helps not only fight off bacteria, viruses, things like that, but it can also help stop or prevent cancer from growing in your body, okay? That's what it's there for. All right, so meiosis, this is a basis of sexual reproduction. Every child inherits a unique combination of genes from his or her parents and displays a unique combination of traits, right? So everyone is a mix of their parents, whether they like to think so or not. Excuse me. Again, you gain one chromosome from your parent, one chromosome from your mother, making 23 pairs, 46 unique chromosomes, right? But 23 homologous pairs. So we need some definitions here. So we have somatic cells, which are any cells found in your body that are not sperm or sex cells. So these are all, somatic cells will technically have all the same genetic information, right? So one of your skin cells will have the same genetic information as your liver cell or prostate, vice versa. They just have differentiation or expression of different genes that make up your different tissues, right? But all your somatic cells have the exact same genetic information. A karyotype, a display of micrographs of the metaphase chromosomes of a cell arranged by size and centromere position. Okay, and we'll see a karyotype right now in a little bit. Homologous chromosomes, so two chromosomes that make up a matched pair in a diploid cell, right? Homologous chromosomes are the same length and contain genes of the same characteristics with corresponding loci. And a loci just means location, okay? Sex chromosomes, so these are the X and Y chromosomes, determine whether an individual is male or female in mammals. So autosomes are chromosomes not directly involved in determining of the sex of an organism, right? So these are going to be all the other chromosomes that we have that are not X and Y. Oh, excuse me. My throat is not feeling good. We end at 8:10. I probably should just stop here, actually. Yeah, we can get through this next time. All right, so the lab today is going to be from photosynthesis. We didn't get to the Mendelian genetics, so we'll get to that next week, which I do apologize for, but you know, things happen. So don't worry, we'll catch up. We have I have a lot of room in the semester to where we can catch up. So do not worry, but we'll do, what was it? Photosynthetic pigments. Okay. So for this lab, you're just going to go through and it'll kind of talk you through light. Visible light is going to be the main source of what excites those photosystems, right? So it's not any other type of radiation. We're talking about visible light with plants, okay? And it's going to just be going through what occurs. Shift control. What is it at? Oh, it's Control Alt. Oh, Control Alt K. Okay. So essentially what you're doing here is you're going to look at the different weights of the chlorophyll, and you're going to look at the pigments that can go up through this, what is called a chromatography paper. Sorry. So what this does is it separates out the different pigments that you'll find in plants. And essentially what you're looking at is just the different pigments and then the weights of the different pigments. Okay, so you're going to see the carotenoids, chlorophyll A and B. Chlorophyll A and B are going to be the main pigments that you'll see that we've talked about in the photosystems, right? You have xanthophylls as well, and carotenoids or carotenoids. And these are just going to have the different, they're going to utilize different wavelengths of light and still provide some source of energy, some source of energy for the electron transfers, okay? But essentially what you're doing is just looking at the different pigments and kind of getting more familiar with the different pigments associated with photosynthesis. So should be a fairly quick lab. Again, quick 5 points. I'll post some questions later about this as well. Don't worry, next week we're just going to hit Mendelian genetics very, very hard. And we'll get into that. That's going to be kind of those big labs that you'll see, Mendelian genetics 1 and 2. And that is a key thing you need to know from this class. So we'll go into a lot of detail about that next week. So just be ready for making some Mendelian charts and working with genetics a little bit. So we'll be talking about like inheritance and Mendelian genetics and X and Y and what the kids are going to end up being and sex linked or inherited traits. Okay, so we'll finish up that lecture and then we'll get right into the Mendelian genetics next week. Okay, any questions? Nope. Okay, well, if not, I will see. All good? All right, thank you, Orlando. If not, please let me know. I know I had some people with issues with the test. Please email me if you have any problems. We'll go over that exam next week as well during lab. Okay, so we'll have like a formal review of that exam and everything that, all the answers. And if you have any questions or you have anything that you want to contest about that exam, you let me know and we'll talk about it, okay? Also, I'm going to be changing up the questions. So instead of having like the quizzes, I'm going to start doing like practice questions instead. So you'll have multiple attempts to take oops, I'm not even sharing, huh? So instead of having these like quizzes that will lock you out if you get them wrong after the first time, you can, I'm opening these up to be taken multiple times. Okay, so I really do want you to study and practice. And I've seen that there's a lot of like really low grades with the exams or the quizzes. So I'm going to have, I'm going to start switching them to these questions instead, unless it's something that you do need to know or that I'm going to just test you on. But for the most part, if you see this little dumbbell, it's going to be practice. So you should have multiple attempts now. If you're having issues with this where it tells you or blocks you out after the first try, send me an email and I'll open it up again or I'll try to play with the settings. But this should let you take the questions multiple times until you get it right. Okay? So there should be no reason why anyone's not getting full points now. So you can spend as much time as you want. If you have any questions about a question, send me an email and I can further explain it to you, or we can cover it in the lab. Okay, so lab is for that too. If you have any issues with these tutorials or things like that, just let me know and we can talk about them during lab. We have plenty of time. Okay, I know lab's kind of time is set up for you to go and do your work, but it's also time for you to ask questions and let me know if you have any issues with the online resources. Okay. But for now on, I'm gonna try to only make practice questions. Okay, so you should have multiple attempts to take it, and it'll accept your highest score. Okay. All right. And that's it. I will see. Well, you can show up for my office hours tomorrow. Just send me an email and I'll send you that link. But again, it's for my micro class, but no one's been showing up, so. If you want to show up, you're more than welcome to. Okay, but if not, I'll see you next week.